Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosynta Consultants in Oakland, California, and the coordinator of the webinar series on behalf of Certip and ESTCP. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to develop predictive corrosion models designed to estimate the functional life of protective coatings on various DoD assets. We will feature two speakers today. First, Dr. Justin Bissinger will present his team's research into the life cycles of various bacteria and fungi and their impact on protective coatings. Second, Dr. Charles White will present his team's work into common molds found on Navy ships, the effects of mold on protective coatings, and potential mitigation options. Each of the two presentations will be followed by a brief Q&A session, and we will conclude the webinar with a longer Q&A session. The next several slides provide instructions on optimizing your webinar experience. If you have not downloaded Zoom yet, please do so at the link shown here and provided to you in your webinar registration confirmation email. If you cannot download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you have technical difficulties or your screen freezes, try keying in Control and F5 to perform a hard refresh of the browser. If you plan to access the audio through your computer, click the arrow next to the Join Audio button, select a speaker and microphone, and follow the prompts as they appear on your screen. If you continue to have difficulties, call into the conference line shown here or submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. You can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email as another way to follow today's proceeding. Also, you can go to the Certip and ESTCP YouTube channel at the link shown here where we will be live streaming the webinar. Note that today's broadcast will be listen only. You can submit questions using the Q&A box on your screen. You do not have to wait until the Q&A period to submit your questions. We do encourage you to get them in as soon as you're able. And when you do submit a question, please add your organization name at the end so that we can identify it during the Q&A session. With that, I would like to introduce Dr. John Lascala, who is the Certip and ESTCP Program Manager for Weapons Systems and Platforms. Prior to joining Certip and ESTCP, John served as the Chief of the Manufacturing Sciences and Technology Branch in the Weapons and Materials Research Directorate of the DEVCOM Army Research Lab. Throughout his career, John worked on bio-based thermosetting resins for adhesives and coatings and environmentally friendly polymers for composites, adhesives, coatings, and additive manufacturing applications. John is the recipient of numerous awards for his work on polymers, including the 2013 Presidential Green Chemistry and Engineering Challenge Award for Renewable Thermosets and the 2010 ESTCP Project of the Year. John, please proceed.
Thank you, Rula. I'm happy to welcome everyone to today's Certip and ESTCP webinar. The next few slides provide a quick overview of Certip and ESTCP. Certip is the Strategic Environmental Research and Development Program established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. Certip's mission is to identify and address high-priority environmental science and technology. Certip funds both fundamental research as well as advanced technology development that ultimately impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under Certip or other research programs and are designed to transition technologies out of the laboratory and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. Certip and ESTCP are complementary programs, with much of Certip research occurring at the lab and pilot scale, with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting lab efforts are conducted. Next slide. There are several environmental drivers for the type of work funded by Certip and ESTCP, with the underlying objective of sustaining DoD ranges, facilities, and operations. This, as you can imagine, is a broad undertaking. It takes the form of looking at maritime sustainability, threatened and endangered species, climate impacts, unexploded ordnance and munitions constituents, as well as other environmental drivers. One key environmental driver is the reduction of current and future environmental liabilities. This involves addressing contamination from past practices, including impacts to groundwater, soils, and sediments, UXO contamination, and developing management approaches for contaminants of emerging concern. The second part of this is pollution prevention, with a focus on eliminating chemicals of concerns or hazardous materials in manufacturing, maintenance, and operations of our installations. And really, today's talk has a bit of focus on that particular area. Not directly, it's really a bit of an indirect aspect, although some of the aspects, we certainly want technologies that remediate microbial growth on coatings. We want these to be environmentally friendly. But some of this is actually due to the fact that we have more environmentally friendly coatings nowadays, as well as cleaning products that may not be as damaging to microbial growth. So it's kind of a little bit of both. We certainly need the environmentally friendly products, but we also have an issue with well, I'm forgetting the words. Anyway, moving on in terms of that. So next slide, we have several main focus areas of research and demonstration at Certip and ESTCP as shown here, including PFAS and AFFF, munitions remediation, sustainable energetics, and various other technology areas besides the ones that we'll be talking about today. Next slide, please. As mentioned earlier, technology transfer is very important within Certip and ESTCP programs. Technology transfer efforts include the development of videos, training workshops, and guidance documents. The webinar series is a substantial component of our technology transition efforts. Next slide, please. Our webinar series highlights research and demonstration efforts from all of our program areas. Upcoming topics include autonomous underwater vehicle-based structural acoustic sonars for underwater buried UXO detection and classification, effects of phenology on species of management truants from wastewater and various others here. Next slide, please. You can find additional information about upcoming webinars at this link right here. All past webinars are archived and can be accessed using this link. Next slide. Please save the date for the 2023 symposium, which will be held between Tuesday, November 28th and Friday, December 1st in Arlington, Virginia. That's always the week after Thanksgiving. And finally, I'd like to remind you that a copy of the presentation of today's session can be downloaded from our webinar page. I would appreciate it if you would please take a moment to complete the survey that will pop up on your screen at the end of the webcast. And I hope you enjoy the webinar today. And with that, I'll turn it over to our first speaker.
Great, thank you so much, John. It is now my pleasure to introduce our first speaker, Dr. Justin Biffinger, who is an assistant professor in the chemistry department at the University of Dayton. Justin has 20 years of experience in fundamental and applied research in bioenergy and polymer biodegradation mechanisms. And before joining the University of Dayton, he served as a research chemist at the Naval Research Lab in Washington for 9 years, where he worked on or led several programs in harvesting energy and materials from the environment using bacteria and fungi. Justin has co-authored over 50 peer-reviewed manuscripts in chemistry and applied microbiology. He earned his bachelor's degree in biochemistry from Ithaca College, his master's degree in chemistry from Bucknell University, and a doctoral degree in chemistry from the University of Nebraska-Lincoln. Welcome, Justin. Please go ahead and get started.
Thank you for the kind introduction. My presentation today will be split up between the following sections, and as a result of a multidisciplinary team between the University of Dayton, the Naval Research Lab in Washington, D.C., the Air Force Research Lab at Wright-Patterson Air Force Base, and Central State University. The biofouling and degradation of protective polymeric coatings continues to be a major issue for the Department of Defense, as seen in the images on the left taken by AFRL scientists from inside of a cargo aircraft. The arrows indicated here also indicate where we removed and isolated some of the organisms used for this program. Interest in the mechanisms these organisms use to survive on these coatings are also valuable as we start to think about plastic biodegradation and the sustainability challenge it presents for generations to come, as shown in the images here on the right of a fungal species degrading mixed plastic sources. And the same mechanisms that are used between these organisms both can be used to accelerate and thwart biodegradation as a whole. However, most renderings of polymer biodegradation mechanisms that try to combine the polymer and microorganism are typically qualitative in describing what are thought to be the essential steps of the whole process. Unfortunately, this leads to mechanisms like the one shown here from an excellent review in 2015 by Mahajan and Gupta, give the impression that the biodegradation that occurs on day 1 is the same as what occurs on day 30. This cannot be true based on how polymers, organisms, and environments change as a result of the colonization of the surface. So as was shown in step 1 of the previous slide that states that cells must adhere to the surface to degrade it, really only cell contact is required for biodegradation to occur. And in many ways, the cell doesn't have to be directly attached to the surface. It could be attached to other organisms in a mixed species biofilm. And so as is seen here in this really wonderful figure describing 5 different ways in which we try to stop biofouling, we can either do it physically, which are described in these A, B, and C sections here, where we're physically keeping the cells or stopping the wettability of them, or we could use biocidal mechanisms that basically kill the cell if they attach to the surface or in the local environment. However, life eventually finds a way around these countermeasures so far over a long enough timeline. This ultimately leads to trade-offs when considering new coating formulations for us and our environment. For example, biocides that can effectively be blended into coatings to stop biofouling for a certain period of time can have untargeted consequences for organisms in the surrounding environment and the warfighter. It is these types of trade-offs that we need to minimize as we look to replace environmentally unfriendly antimicrobial and anti-corrosion coating additives with more environmentally friendly ones. This change could increase the amount of biofouling and ultimately biodegradation that we observe in the future, because the compounds that we're using in order to become more environmentally friendly are not aggressive as the ones that were not environmentally friendly. Since biofouling is inevitable over a long enough operational period, and biofouling can lead to coating biodegradation, can we identify how much of the resulting biodegradation of a coating is driven by a microorganism or collection of microorganisms, or just initiated by them? This was one of the major focuses of our program. Can we define, using biofilms of environmental fungi isolated and identified by AFRL scientists from degraded coatings inside of cargo aircraft, how much of the biodegradation process requires an active microorganism or their cellular components? And in this case here, we're just breaking out the fact that at the beginning of degradation, you have cellular attachment, you have potential secretion of these hydrolytic enzymes, and their activity and the cellular activity don't necessarily have to be coupled, and that there's a time-dependent component to it. So where does biology stop and chemistry takes over is a key question in order to address biofouling with more environmentally friendly additives. For this presentation, we are only going to discuss 2 yeasts isolated by AFRL scientists, Papillotrema laurentii and Naganitia albida. They were identified as being hydrolytic using a colloidal polyester from Covestro called Impronel, which is a good qualitative screening compound for hydrolytic activity, as is shown in the images below. However, these images also should give researchers some caution that of these hydrolytic enzymes, clearing of Impronel is not a direct measure of all of the different types of hydrolytic activity. It's just a really good qualitative measure if you're looking to screen. These 2 yeasts, as we go about the presentation, are phylogenetically dissimilar, but both hydrolyze polymer coatings rapidly, enabling important comparisons between the molecular and cellular levels. So here is a phylogenetic comparison of the 2 organisms. They're phylogenetically dissimilar, and yet they seem to hydrolyze coatings of equally. So what types of mechanisms are they using? We compared their hydrolytic activity as biofilms on aliphatic polyester polyurethanes that would release succinate and adipate if hydrolyzed to a commercial polyester polyurethane called Irogran on the right here that is more recalcitrant, that is a more recalcitrant coating to degrade, but releases adipate when hydrolyzed as well. The release of succinate and adipate from these coatings was one way that we could compare the responses of the biofilms, since succinate stimulates growth and adipate is a viable, though harder to use for replication, carbon source. We analyzed the biodegradation using several overlapping analytical techniques in this program, with IR spectroscopy and microscopy being the most flexible approach. The NRL team on this project even fabricated a high throughput screening mount to increase the number of replicates and data points in a single series of experiments. And the approach that they used is shown here with the custom mount that Dan was able to fabricate, and the resulting data that then resulted from that, showing clear isospecific behavior of the carbonyl being hydrolyzed and the acidic degradation products being formed. Using this approach, we compared the rates of hydrolysis of each coating with Papillotrema laurentii and Naganitia albida. In this particular case, we're just showing the results for Papillotrema, but the Naganitia results were very similar. We observed that most of the coating hydrolysis occurred within 4 days of spotting the cells on the surface, with the remaining hydrolysis occurring slowly over the next 10 to 15 days. We also observed less degradation with the harder coatings, or more crystalline coatings, the PEA and the Irogran coatings, which is consistent with their increased crystallinity compared to the polyethylene succinate coatings, which are shown here. These data confirm that most of the hydrolysis occurred initially, but the remaining degradation over 10 to 15 days suggests that another mechanism was operative after the initial 4-day period. And of course, on the polyether coating, which is just a polyether top coat only, polyether polyurethane top coat, we didn't see any degradation or hydrolysis at all. Also, within the first 4 days, we see the highest rate of cellular activity based on the headspace gas analysis of CO2 during the degradation of each polymer. So down here shows the representative vials of how we performed this experiment. This one here had the Irogran polymer coated on the inside with water. And this vial over here had Irogran with a culture of cells that have been washed with water of the Papillotrema laurentii after 22 days. You can see here that the coating has been hydrolyzed here. There you can clearly see into the vial. And as a result, we were able to sample the headspace. And what we observed, interestingly, is that the differences from each of these organisms we'd observed differences between each of the organisms on the individual coatings, with succinate coatings producing the most amount of CO2 for Papillotrema and the adipate coating for Naganitia. These results suggest that though these organisms are degrading polymers with similar rates, their responses to the polymer hydrolysis products are certainly different. So we observe that cellular activity slows down as the polymer is degraded, but were these cells viable at that point? We developed a method using a fluorescent viability stain, FUN1, it's a commercial stain, shown here that enabled the measurement of cell viability of a biofilm over time on each of the polymer coatings during the biodegradation experiments. Red cells are viable, green cells are not, and by calibrating the red to green ratio, we determined that the viability of the cells over 25 days. And the results are shown here. These results are actually consistent with the CO2 results that even as the coating is being actively degraded, and of course we expect the cells to be actively respiring during that period, the viability of the biofilm is actually dropping. And only on succinate-based coatings do we observe approximately 30% biofilm viability after 25 days. And on the other coatings, we observe almost no viability of the biofilm. So what is this drop in viability? We analyzed the papillotrema cells incubated for 30 days. So we went to the end of our experimental time period on each of the polymer surfaces with no supplemental nutrients and monitored the data monitor the changes by transmission IR and IR spectroscopy. What we observed on the coatings was that a dipate-based coating stimulated protein catabolism, shown here by the decrease in the amide peak that we're seeing here using this amide peak fitting, which is representative of the protein-based components of that biofilm. While the succinate, and so that was for a dipate, but while succinate coatings are exposed to cells, they do not initiate catabolism of the protein, shown here by no change that we see with the succinate coatings, even though the viability of the cells is decreasing during this time period on each of these coatings. And if no degradation is observed, so in this case on these ASP 108 coatings, which are polyether based, we see no catabolism of proteins, even though the cells are most likely looking for a nutrient, but rather these cells, if they are not performing hydrolysis, they enter a dormant state or quiescence. So nutrient limitations are expected. So nutrient limitations should be expected under field relevant conditions. And what we observe from these cells due to starvation is the induction of autophagy. And so we are using an autophagy stain. We're seeing inclusion bodies within the cells. We're seeing all of these indicators that these cells have entered autophagy. And autophagy is a natural conserved degradation of the cell initiated by stress and ultimately leads to recycling of cellular components of most eukaryotes. So the catabolism of protein observed from the loss of the amide signature and the IR data before confirm that with the successful hydrolysis of the polymer, but release of a poor carbon source like a dipate. So here we're talking about the cycles that are occurring as the polymer is degrading and the monomer is coming back into the cell. The cells were stressed and entering a state that would allow them to recycle the cellular components in order to survive. However, if a viable carbon source is present, like succinate, the same catabolism is not observed, and that carbon source is being used for energy and ultimately growth. Well, we also confirm this autophagic state using RT-qPCR through a lot of really hard work by our AFRL teammates. And what they used was the ATG8 gene, which is a key autophagic protein identified in yeast of approximately 34 genes. We observed as early as 30 minutes after spotting the cells on every polymer surface, the upregulation of this gene and the upregulation of a cutinase gene, which that we have now identified as being the primarily hydrolytic protein secreted by Papillotrema laurentii. So even after 30 minutes, we were able to, we see responses that are indicative of nutrient limitation and ultimately autophagy, which is leading to then this hydrolysis. And over here, this is showing a description of all the different genes or how ATG8 is involved. And this over here is the spotting of the different biofilms onto fluoro dishes of each of the polymers, showing the representative of how we approached this experiment. This was looking at the fold change of biofilms over, in this case, it was 30 minutes, but we also have data extending out to 25 days. So Based on the collection of these results for Papillotrema, we created this summary table which highlights our analysis. In general, if there were no usable carbon sources, the cells go quiescent. So that would be the case here where we're seeing non-hydrolyzable polymer, we're seeing cell quiescence. No nutrients, cells become dormant. For succinate-based polymers, so ones in which we can now hydrolyze the polymer, the carbon source is a viable nutrient source, and we see no signs of catabolism, but yet we do observe degradation and autophagy occurring of the cells. But for adipate, we observe cell death and protein recycling, but for some reason that we're currently looking into, we don't see quiescence, which up until this point is unsolved for why we don't see the cells become dormant when they're receiving a carbon source that isn't as good of a carbon source for survival. So if the majority of cells are not viable after 4 days, what exactly are these organisms using to degrade these polymer coatings, and how are the cells using them and utilizing them? So what we did was we compared the secreted proteins after being concentrated in nutrient-rich broths, isolated from both Papillotrema laurentii and Naganitia albida. And so here we show 2 different electrophoresis gels. This gel over here is being stained with just a simply blue Kamasi stain, and we're showing the different protein fractions that we have. And then we stained the exact same, we ran the 2 gels in parallel, and what we had is a second gel that is now only going to show hydrolytic proteins in the gel. And so in this case here, we see this really stark difference between Naganitia albida and Papillotrema, where we see this 22 kilodalton hydrolase here at pH 4 and pH 7.6. But for N albida, we don't see something of comparable size, but rather a rather large protein that is approximately 170 kilodaltons in size. So there's only, at this point, we only have 1 active esterase in either of these 2 modal yeast. And so they are both non-modal. They have to be moved on the surface, but yet they're using 2 very different hydrolases. And so now we're going about, and we've already isolated the hydrolase for the Papillotrema strain, and we're really aggressively going after the Naganitia strain. Now, the reason why this is important and how it links back to ultimately the bioenergetics of this entire process is that so what we see with Papillotrema is that we see strong cellular attachment to the surface. And so here are a variety of time-dependent bright field images of Papillotrema cells being spotted onto the polyethylene succinate coating. And what you see here is basically the same pattern across all 7 days. However, when you look at Naganitia, what you see over time is the pooling of these cells gradually with the water that's condensing on the surface. Something that I was remiss to say is that all of these experiments are being performed at a relative humidity of 95%. So in other words, we know we're condensing water at this relative humidity. And so having water condense on the surface is not something that's foreign to what we're observing. So in this case here, we have for N albida, we have 140 kilodalton protein and a cell type that's being loosely attached to the surface. And so the larger the protein, the less likely it is it's going to be able to diffuse into the polymer, and more likely an organism that wants to be moved by pooling water would want to keep the hydrolase near it. And so that would be our operating hypothesis, that basically the large hydrolase is being kept near the Naganitia albida because it wants to keep it near it, as opposed to Papillotrema laurentii, which secretes a small, very active hydrolase to cells that are strongly adheres to the surface. And so this difference between the sizes of the hydrolases and how they're being used leads to a really interesting connection to the bioenergetics of this entire process as these cells are trying to survive over the period of time. Now, as I said before, AFRL has done exhaustive work in order to identify the actual cutinase from Papillotrema laurentii. So what they've done now is they've created and cloned and expressed the putative recombinant cutinase using E. coli. Here, they're showing the different fractions from the FPLC, and of course, the clearing of an impernyl agar plate assay showing that these enzymes are still hydrolytic. Also, researchers at AFRL have now taken that sequence and used AlphaFold in order to come up with the potential structure of this cutinase. And we see here that the cutinase has no lid. And as I said, the predictive structure was based on AlphaFold. That is, based on using AlphaFold. And of course, the structure can now be compared to other hydrolases and cutinases in order to come up with structure function comparisons. And this is exactly what we planned for the Naganitia hydrolase that we can isolate in the near future. And down here, we just show a variety of soluble esterase activities. And we see here that we have activity of the wild type strain for the hydrolysis of the soluble hexanoate paranitrophenol derivative. And over here, we've also now looked at several different lengths of chain length, and we see the hexanoate actually ends up leading to the fastest amount of, or the most amount of hydrolysis. In addition to this, you need water in order to hydrolyze polymers. And what we've constantly been dealing with this project is that these organisms actually secrete deliquescent materials in order to condense water at relative humidities well below the where water should condense. And so this is an example here where we show a control supernatant that did not have cells grown in it. And at a relative humidity of 60%, we see that the fungal supernatant clearly condenses water. And this was a constant problem that we had throughout the project. It's a good problem, by the way, that we were trying to get rid of the water to take our measurements, but really it's a really fascinating bioenergetic advantage for organisms to be able to condense water at relative humidities where they wouldn't necessarily have pooled water to perform the search for food. And so deliquescence also links to autophagy, which also links to necrosis. And so I think this is a really excellent image from a paper in 2018 that sort of starts to describe how autophagy, as it's related to survival, can also be related to this cell death, right? This entire presentation, we talked about how the cell viability kept on dropping and dropping and dropping, and how this autophagy state ends up being upregulated as a way for the cells to survive. So degradation was really being linked to cell death at the towards the latter portions of the mechanism of the process. So in conclusion, the cellular activity, we identified cellular activity on different coatings over 7 to 10 days. We see with these two particular organisms that they're entering this autophagic state as a result of nutrient limitations. We've now isolated identified a novel cutinase responsible for the hydrolysis of polyester polyurethane coatings, and we're searching for new ones as well. And of course, the key biogenic components that we're using for modeling is also how we're addressing are being determined. The benefits to the Department of Defense is that we want to create a more quantitative approach to understanding the mechanism. We're describing this as our bio-life cycle analysis shown here, where basically we want to get away from using these sort of qualitative pictures and start to think about this as inputs and outputs and bioenergetic balances in order to approach biofouling and predictive type models in the future. All of our life cycle inventory and overlapping data sets will be publicly available. Of course, this idea of deliquescent materials also has a huge application for trying to condense water in arid climates. And of course, the novel organisms and enzymes isolated are applicable to plastics degradation and the valorization of waste. None of this would be possible without my collaborators and all the excellent and talented people that have been part of this project for now greater than a decade on all the different things that we've done together. And these are the figure sources. And that's it. I'll take questions.
Great, thank you so much. As a reminder, if you guys have any questions, please do submit them in the Q&A box. For now, we've received several that we're going to go ahead and relay to you, Justin, with the first one being, what are the next steps in your research or work?
Oh, the next steps would be taking all of the analytical data we've taken over these different time periods, and then basically using that information to create these bioenergetic, basically looking at the free energies all the way down to protein synthesis and secretion to understand where does this process start to tip in our favor in the sense of stopping biofouling, or in the organism's favor that they can continue to degrade and foul our coatings. So bioenergetics is next for us.
Great, thank you. have you tried to quantify how frequently these two yeasts or similar microorganisms are responsible for coating biodegradation within the DoD services?
Oh, I see. Like their abundances within other weapon systems. No, we haven't done that, but that would be really interesting to do. I know that my AFRL colleagues, they have done really exhaustive metagenomics of several different, let's just call them weapon systems, that they are looking at how prevalent these organisms are and where they're being found. But in other services, that would be really important data.
Great, thank you. Can you elaborate on one of the applications of the results collected during your research?
Oh, certainly. Yeah. So really what one of the applications would be, and I mentioned it in the first comment as well, is the biological life cycle. Like I really think it's important for us to now think about biodegradation in terms of different time periods and phases. And within those phases, degradation doesn't have to be the same. And trying to create adaptive models that have that within it will be important in trying to design coatings that can predict how biodegradation is going to either occur or not occur in the future.
Thank you. Is it possible to separate the condensation of water from the fungal secretions or the changing surface potential of the degraded polymer?
Yes, but you can't perform that experiment with the live cells. It really is challenging because as the polymer degrades, you basically release these more hydrophilic functional groups, and as a result, they change the surface potential of the polymer. That can start to then condense water. And so trying to come up with a balance between what is coming from the organism and what's coming from the actual coating being degraded is one of the big challenges that we are continuing to address.
Great, thank you. This next question is from the Naval Surface Warfare Center, Dahlgren Division. Have you looked at hydrolysis of the two cultures in co-culture or with other organisms that may have been identified on similar coatings?
Yes. I wish I had a better, yes, we have started going down that path. We were more focused when we started doing the co-culture work. We didn't feel like challenging Naganitia with Papatrima was what we wanted to go after, but there are other organisms found in these naturally occurring consortia that we are interested in because they have the ability to oxidize alkanes. And as a result, the connection between the oxidation of an aliphatic polymer with its eventual hydrolysis with these organisms could lead to an interesting synergy and effects. It's a great question, and yes, we are pursuing it, but not with these two organisms together.
Thank you. This next question is from Raytheon. How challenging is it to reproduce the full range of relevant real-world environmental conditions in your current test beds?
Oh, real-world conditions, yeah. So for what we're doing, right, there's two ways you can go, right? There are the field environmentally relevant studies with mixtures of organisms that ask one type of question, but that question is not what we're focused on. What we're focused on is trying to control every variable and trying to come up with, from a fundamental point of view, what does the organism want? And that question requires removing basically all other variables out of the equation to come up with like what was the most important component to the organism from a bioenergetic balance and what ultimately occurred to the coating. I totally understand like why you're asking that question, but this program was not focused on trying to get to relevant field conditions.
Thank you so much. How do temperature and humidity affect the growth of these yeasts and the resulting biodegradation of the coating?
Right. Yeah. So it ends up. Actually, I can't say this. So okay, I'm going to go with that is a wonderful question. There is a temperature dependence that we're observing, but because of where we are with that experiment and what's been cleared, I won't be able to answer that one. But it's a great question.
Thank you. Did you and your team find that working with only biofilms is challenging?
Yes. Yes. It was when you deal with, I mean, and anyone that's done this type of work, like when you deal with biofilms, you have very little sample and a lot of just of the good bread and butter analytical techniques and proteomics. You just don't get enough sample to work with. You don't get enough sample in order to come up with any reasonable analysis. So like trying to look at what proteins are secreted into a polymer coating, you would have to have a very, very thick coating and a very, very active organism. And so working with biofilms, it was amazingly challenging for the team. But when you work with federal labs and such talented scientists with these really different scales of analytical techniques all the way down to AFMIR, it becomes a little bit easier.
Thank you. How does biodegradation continue on the coatings if the cells are entering autophagy?
Right. So if you think about that question, that's actually leaning towards, well, how active are these enzymes? Like over a certain period of time, is there an expectation that that enzyme should become deactivated? I think there should be, but is it completely deactivated? I don't know. So why is degradation still occurring? Well, that could be the fact that because we've initiated, the organism initiates degradation, but ultimately in the end, it's that initiation that then leads to these sort of autocatalytic cascades of both chemical and potentially biochemical mechanisms that ends up degrading the polymer slowly and continuously throughout the lifetime of the polymer. It's a great question.
All right, and one last question before we move on to Charles's presentation. You mentioned that you ran your tests in a closed chamber. Would you expect prolonged activity and delayed autophagy if you had used, for example, another setup like a chemostat setup?
Oh, chemostat setup.
This is a question from the Pacific Northwest National Lab.
Right. Yeah. So we're so, because everything we did was biofilm based. The idea of using a chemostat, though provocative, I think it wouldn't fit with the air solid interface that we were trying to understand. But certainly, if you were doing, for example, looking at coatings that are constantly submerged, like on the hulls of ships, then certainly you could use a chemostat culture and then flow that out to some sort of cell holding the coating and look at degradation that way. But for us, we were interested in air solid interfaces and not submerged coatings.
Great, thank you so much. This was really engaging. We'll try and get back to any questions that we haven't answered towards the end of the webinar, but at this point, we're gonna switch gears to our second speaker, Dr. Charles White, who is the Senior Coating and Corrosion Technical Advisor at the Corrosion and Coatings Engineering and Sustainment Branch at the Naval Surface Warfare Center, Carter Division in Maryland. Charles has worked as a coating engineer for the Navy and the Marine Corps since 2016. And for the last few years, he has been leading a collaborative project on mold inhibitive coatings and fleet surveys to identify and assess the biodiversity of fungal species on U.S. Navy surface ships across various U.S. Locations and temperate zones. Charles' research interests include functional coatings and stimuli responsive coatings. He received his bachelor's degree in polymer science and high performance materials from the University of Southern Mississippi in Hattiesburg. and master's and doctoral degrees in chemical engineering from Auburn University. Charles, please proceed.
Good afternoon. Thank you so much. I'm Dr. Charles White from NSWC Carter Rock, and I'm very excited to present on behalf of myself and my project team. We're very proud of this project and very excited by both the progress and the feedback we have gotten over the past 2 years from the technical community and the fleet. And I want to thank everyone for being willing to spend their lunch hour to look at pictures of mold with me. I also want to thank ESTCP for the invitation to present at this webinar today. So I wanted to go over with you today that the impetus for our research project and what we have learned about the occurrence of mold throughout the NAVSEA fleet. Mold and fungus growth is not typically reported as part of the ship corrosion and maintenance reports, and has to be relatively extreme to be noted on the maintenance report cards. As we've peeled back the layers of the onion on this project, we have received a lot of feedback and interest from the fleet and ship personnel to really interested in finding a proactive long-term solution for fungal growth that has really been underrepresented in our understanding of uh, ship life and operations. This has uh, included surveys of the fleet to document mold growth and really understanding what the standard operating procedure for ship's force for palliative or temporary cleaning, and understanding what the representative examples of fungal growth and the results of the DNA survey across the fleet to determine commonalities between fungal growth and assist in the identification of the most effective cleaners and procedures to prevent fungal growth in the future. We've worked with an industry partner to formulate an inhibitive coating technology into a mil-spec qualified coating that I'll tell you about, and have begun performing in-house conformance testing for the prototype coating, and have performed ship demonstrations. Fungal growth can be found almost anywhere where moisture is present, as anyone that owns a shower stall or operates in a high humidity environment can attest to. And what we have found in our research has been that it's almost impossible, particularly on ship growth and on DoD assets, to separate the term dirty from fungal growth. And because they are typically, if a surface is dirty, there is fungal growth, either macroscopic or microscopic. Conventional solvent wipes and standard operating procedures available to the to fleet personnel are not capable of completely eliminating all spores and hyphea from the surface of the ship surfaces, and can actually exacerbate regrowth of the fungal in as short a time as 2 weeks after cleaning. Areas prone to fungal growth are all over the ship, and these include areas that are exposed to hydraulic fluid or other organic fluids, exterior air and salt spray, and just areas that are challenging to clean and will store high degree of cardboard or other materials, where fungal growth to provide shelter and food for fungal growth in the future. And areas on ship that are challenging to keep dry due to the confined nature of the ships, out of reach areas, or the blockage by piping and other structures. This impacts fungal growth for a variety of reasons and motivates us to want to find a permanent solution, because fungal growth, all mold growth has the potential to negatively affect personnel respiratory health due to the presence of spores in the air and in particular, the mitotoxic fungal growth can be a particular health hazard for ship's force. In addition, fungal growth can be a repetitive cleaning and uh, require repeated action by ship's force that takes their time away from other other maintenance activities. It can exacerbate corrosion or breakdown of coatings, leading to corrosion, and become a reinforcing cycle where reduced coating performance and protection results in mold growth and reinforces each other in a synergistic manner. This is a problem because fungus is a relatively difficult microbiological to control. The graph on this slide shows the general relationship between microbiologicals and their resistance to common classes of liquid cleaners. Fungus highlighted in red is moderately resistant to different microbiological growth and cleaners. This is important because there are two really important aspects for controlling fungus growth, particularly on high risk areas such as ships. You have to clean both the macro growth and the hyphea, or the spores and other micro growth present on the different surfaces. Otherwise, if you just clean the macro growth and spread around the spores and hyphea, it can exacerbate growth and spread out the fungal growth over a larger area and exacerbate your problem rather than cleaning it. And this, want to thank Lysol and Reckitt, their personnel, for letting us leverage this slide for the graph on this slide from a previous ESTCP presentation. In our surveys, ship's force were interviewed to determine the standard operating procedure for fungal cleaning. In most cases, severe fungal growth is directed by ship's force to seek out contractor remediation, which can have a very, very long lead time for ship's force, including a year or more for the more severe cases of mold, and maybe just a temporary reactive cleaning. Most commonly for minor outbreaks, ship's force are instructed to use organic solvent liquid cleaners or dilute bleach solutions. And these cleanings, combined with mechanical wiping, are very good at cleaning the macro growth, but are largely ineffective at cleaning the microscopic or hyphea growth and removing the spores, which cause immediate regrowth of the fungus. The effect of these cleaners on the hyphea are shown at the bottom of the slide on the photos of the last three. Those images that look like cracks in the coating are the long filamentous growth or hyphea that will result in rapid regrowth if not removed. Each sample for these pictures was exposed to organic solvent and wiping with little to no effect on the microgrowth, whereas one cleaner, the Nav Clean, was shown to be highly effective against the microgrowth. Nav Clean was developed by NavAir specifically for use on aircraft to remove mold and is available through the Defense Logistics System via Millstrip and has an NSN for ordering. It's a multi-component cleaner that's mixed on site with water, spray applied, allowed to rest for 15 minutes, and then rinsed off with DI water. It's been shown to be highly effective cleaning, but has not been traditionally available for ship's force use. For this reason, we have included it in our ship demonstrations with the potential to implement and transition it to ship's force through revisions of the AEL or the approved equipment listing, and the ship's hazardous material listing, or SHML, for use across the fleet. And we've gotten high interest from the fleet and support from Ship Command for transitioning this cleaner. Other common cleaners, as I said, are typically seen through either a organic solvent or a dilute bleach solution, which really are any liquid cleaner once is a reactive measure to fungus growth and is only used after fungus has reached a certain point to cause a cleaning. Other methods such as NavClean have been shown to be highly effective. Bleach has been shown to be a risk for corrosion on certain surfaces, and the organic solutions and bleach have been shown to exacerbate cleaning if not done with extreme care. More sophisticated techniques such as UV cleaning or steam cleaning typically require either more expensive equipment that's not available to ship's force, more skilled labor, or some amount of downtime that is not conducive to the ship's scheduling. For this reason, the cleaners have been described to mold growths have been described to us by ship's forces almost a way of life aboard ship or just a cost of doing business when the ship is on ship service with no real proactive preventative solution. And if I want, the last takeaway I want from this slide is highlighted here at the bottom. Bleach solutions are not the answer. By and large, this has been the most common answer to our surveys for standard operating procedures or what is available to ship's force to use. The bleach solutions are again very good at cleaning the macro growth, but will spread and exacerbate growth unless a certain concentration with a certain residence time is reached and can clean the hyphea from the surface. Example of taken is of mold growth on a destroyer is provided on this photo. This is a bulkhead from a destroyer in Norfolk that is subject to occasional condensation exposure, where the original color of the bulkhead is shown on the right-hand side of this soft white material. And can be seen on these cross pipings here as the most representative color. Any darkening behind that is mold and gives you a relative idea of representative of mold growth on this surface. Similar for overhead space on a L-class amphibious ship that's can be exposed to exterior air and other fumes and exhaust from vehicles will collect on the surface, and you can see the original color is represented by this pipe here, or this the bright white here presented here on the right. Any darkening or occurrence beyond that is mold growth. As you can imagine, this has a potential health effect and respiratory effect on ship's personnel, and the effect on their morale, because these ships are home to the sailors for 6 to 9 months out of the year, and inevitably will have an effect on the ship's personnel just from smell and morale alone. In addition, we saw on a carrier survey that we did after carrier went out of service, defects were introduced into the paint through service, and fungal growth was seen to grow at the cracks in the paint, where either a follow-up study that we are working on of whether the cracks are actually providing shelter for the spores and hyphea and allowing them, preventing clean, or if the fungus is actually feeding on the coating and degrading the performance and of the coating, resulting to failure of the paint. As part of this survey, we inspected a total of 30 ships, with carriers and destroyers being the most representative of the fleet and the most common ships in the fleet and the focus of our inspection. Across the ship spaces, we surveyed a total of 117, including habitable areas, equipment rooms such as fan rooms, storerooms, escape trunks, and auxiliary access areas, galleys, cooking areas, or storage areas such as weapons lockers and other spaces on the ship. This really gave us an idea of how fungus is growing across the ship and the prevalence of the areas where fungus can be seen and documented. When during the surveys, we made special effort to collect samples from the ship's surveys across ship class, geographic location, and temperate zone, represented on this slide, where the areas of the survey included Norfolk, Virginia, Mayport, Florida, Pearl Harbor, San Diego, California, Everett, Washington, and Yokosuka, Japan. You can see the temperate zones where these samples were collected and where the focus of our efforts went, and representative of where the fleet operates around the United States. I want to give a special thank you to Mr. Michael Kibler of Elsley Technologies for doing some of the swab and biotape samples and taking fungal species selection across these locations and crawling into some of the more moldy and dank areas on the ship for us. So the results of the DNA analysis show some interesting results, particularly for the larger, more high traffic, warm area ports, where there was a high occurrence of mold, but of a relatively lower fungal concentration across the ships, but a higher fungal biodiversity, as represented by these graphs, with the representative and the names of the fungus identified in the above the chart, where we move into areas that are little more low traffic or have cooler areas, particularly in Everett, Washington, where we still saw a high occurrence of mold, but it was a low relative biodiversity of what was occurring, and to the point where we did a barge inspection in Ketchikan, Alaska, and saw relatively no mold in those cold waters in that area. This highlights the importance of selecting the inhibitors for the coating for implementation to address a wide variety of fungus across different geographic locations, and the need to look for commonalities of the fungus across the fleet. In addition, we were able to identify the toxic mold species for the different DNA samples analyzed. Some as high in samples as high as 30% of the sample were toxic molds. So this is a high concern and high motivation for us to find proactive long-term solutions for mold growth. The most common species that we found in our DNA studies are listed on the screen. Apologies, I don't want to embarrass myself through my Latin pronunciation, but the 4 species are pictured below as well, and want to thank the databases for allowing us to leverage photos and representative samples for the growth here. In addition, we performed in situ coating condition analysis, looking for non-destructive testing, including hardness, gloss, color assessment, measurement of the dry film thickness, environmental readings, and a solvent resistance test through MEK double rub, and collected the data looking for the impact of fungal growth on the performance of the coating on ship. The analysis resulted that there was a slight lowering of the coating hardness where mold growth was prevalent, and no noticeable difference in color and gloss, but there was a small difference in color due to the growth of the mold, where areas with high mold growth had slightly darker than the control areas. One of the more interesting correlations that we found was high mold growth tended to favor areas that had thicker average dry film thicknesses than the lower areas. And again, that's a area that we are interested in looking up and investigating in a follow-up proposal to look at causation versus correlation on that. In addition, the environmental lower surface temperatures and more humidity resulted in more mold growth, with the greater difference being between the air and surface temperature with higher humidity and more condensation. There was very little difference in the solvent resistance observed through the MEK double rub between the high mold growth areas and the low mold growth areas. The conclusions were that mold growth is widespread across the fleet and is a widespread issue that needs a proactive preventative measure to prevent in the future. Toxic molds can be found across all the areas inspected and can be a respiratory hazard for ship's force and sailors. And we really don't have a unified method of addressing mold once it occurs, let alone a proactive long-term prevention, where cleaning does provide a benefit, particularly for macro growth, but it's difficult, time consuming, and very temporary, where regrowth can happen in 2 weeks after initial cleaning. Contractor support has a very long lead time, and there is a high support and interest in fungal remediation techniques. Proposed solution has been through the development of a novel mold inhibitive coating and testing of NavClean as a liquid cleaner for ship demonstration. And the coating developed will be a modified version of a qualified MIL-PRF 24596 or a interior fire resistant coating with mold inhibitive additives developed with our industry partner through a proprietary set of inhibitives that were specifically designed to be effective against the DNA results of the DNA analysis and ship survey. During our in-house conformance testing, we are working with the industry partner and with our subject matter expert, Dr. Jason Lee, and at the NRL in Stennis Space Center in South Mississippi to develop testing against 11 different molds identified through the ship survey using different inhibiting components at different levels of loading. The long-term testing will be done in accordance to MIL standard 810H, where fungus is introduced to the material and then to a dish to the coating, and then subject to high growth, high humidity, favorable conditions. And conformance testing will be done in accordance to MIL spec 24596. Then comparative testing with the NavClean versus traditional shipboard cleaners on the 11 selected molds will be performed to ensure high compatibility of the cleaner and support transitioning NavClean to ship's force. So we have performed 2 ship demonstrations in 2023 and are hoping to provide material for additional demonstrations for destroyers and other platforms. The demonstration focused on the cleaning ability of NavClean and the mold resistance of the prototype coating, and has been applied and will do regular assessments up to a year, particularly after ship deployments, looking for prevention of mold regrowth compared to control areas on both CVN 72 and aircraft carrier CVN 76. This is our photos taken from its ship demonstration from the first coating demonstration in 2023 and in January, early February. The condition of the coating as found here at the top, where all the dark areas are degraded coating and fungus, versus the newly painted applied prototype coating, where we got a very high degree of favorable feedback from ship's force from the bright white appearance and the application properties of the coating, and are looking forward to receiving the initial feedback from the ship demonstration over time. In the short term, we have not seen any mold regrowth in these areas where the new painted are, but are looking forward to getting results back after the ship has been deployed and is exposed to high mold growth conditions. Opportunities for growth of this area outside of NAVC. I want to emphasize that fungus and mold growth is not a NAVC specific condition, and these photos were provided by Dr. Jason Lee and by Mr. Andrew Sheets, who's our representative for the Marine Corps. The pictures on this slide include an Air Force helicopter interior where you can see the fungal growth on the different surfaces and the complexity of the surface and how keeping that area clean, particularly when it's exposed to near constant exposure to hydraulic and other organic fluids, provides an excellent growth area for ships and for fungal growth and difficult area to keep clean. In addition, you can see another demo that was done on a helicopter where all the black is fungal growth and this light blue or this pale area is the cleaned coating. So relative high extent of fungal growth in these areas that it's difficult to keep clean. And we again have high interest in developing a proactive long-term control mechanism for fungal growth. In addition, ground vehicles are highly susceptible to fungal growth from cushions and floor foot wells where moisture can accumulate and high humidity conditions can lead to fungal growth, but are difficult to keep clean in the long term. The overall benefits to the DoD from this work are to reduce fungal growth for platforms across the DoD and prevent impacts to personnel, respiratory health, and prevent degradation of coating performance and reduce coating performance on the occurrence of corrosion across DoD platforms. The reduced cost of cleaning in both manpower, time, and hazardous material accumulation and disposal and its impact on the environment are a high benefit to the DoD, as well as the improvement of personnel morale where they have to inhabit or operate platforms that previously have had a high degree of growth of fungus, but by introducing a long-term inhibitive coating can reduce that effect on ship's personnel. With that, I would like to thank my project team, Dr. Jason Lee from NRL Stennis Space Center, and Mr. Patrick Cassidy and Mr. Mike Kibler from Elsley Technology, a contractor here for us here at Carderock, and would be glad to address any questions. Thank you, and thank you for your time.
Great, thank you so much, Charles. We've received a number of questions, but let's start with the obvious one in terms of next steps for the work. Are you done, or do you plan on continuing this effort? And if yes, how so?
So we would like to continue by this work has focused on more practical demonstration of fungal inhibitive coatings. And we would like to do a more fundamental analysis of what's actually happening to the coating as fungus is growing. Is the coating just providing a shelter for fungus growth, or is the coating actually being degraded and metabolized by fungal growth in the long term, resulting in reduced performance? In addition, for follow-up for this current work is to qualify this fungal inhibitive coating to the MIL PRF 24596 and make it available for ship's force procurement and application across the fleet and to the other branches of the DoD.
Great, thank you. And what is the approximate timeline for the next steps you just described?
So we are hoping to get the results of the ship demos within the next 6 to 9 months and use that. And we are working with the corrosion and coatings, coatings and corrosion control tech warrant holder at NAVC to introduce this coating to the fleet and develop a NSN for ships procurement. In the long term, we want to revise the MIL 24596 specification to include a special classification for type and class of material that is specific for fungal inhibitive coatings that can be qualified from different manufacturers to this specification and allow procurement by the fleet.
Great. How much interest or feedback from the fleet have you received to date?
We have gotten a high degree of feedback and interest from the fleet, including from ship's command, including the captain of some of the two of the aircraft carriers who were interested in applying this coating and using the nav cleaner across their ship to control mold growth and keep the ship clean. We have received a lot of feedback and interest from destroyers, the destroyer community as well, and the L class of ships. We are hoping to procure some of this material and provide some of the material out to the different classes of ships for them to do demonstrations as well. And we've gotten a lot of feedback from the different branches of the DoD, including the Army, the Air Force, the Coast Guard, etc., where they have described their experiences with mold and the prevalence of mold on their assets and have experienced and are interested in developing this technology to specifically for their coatings as well.
Great, thank you so much. Can the coating you described be purchased commercially by the Navy or other DoD branches?
That's a very good question. We are working with our industry partner, and due to this being public forum, I don't want to name specific names, but can talk offline on that. It is not yet a commercial product from our industry partner, but they are working on introducing it commercially, and it can be ordered through their material as a custom order if you reach out to their sales rep.
Thank you. This next question is from the Naval Surface Warfare Center, Dahlgren Division. Were ship surfaces cleaned with NavClean before the coating was applied, or was the coating applied directly onto the contaminated surfaces?
So we did not do any of the trials where the coating was applied to a contaminated surface. All the trials were done with a cleaning beforehand. We did do different variations of the application where we did one with a conventional cleaning with isopropyl alcohol as the representative organic solvent cleaning in accordance to SSPC SP1 solvent cleaning. We did a second trial where NavClean was applied prior to the application of the fungal inhibitive coating, and the third trial was done where isopropyl alcohol was applied before was used to clean before the application of the control 24596 coating.
Great. This next question is from the Air Force Research Lab. Were transcriptomics considered when sampling the mold contamination on the different ships? Since the degradation is enzyme driven, finding out information about the more prevalent metabolic pathways could provide a good starting point to learn about the chemistry that could in turn aid in identifying potential inhibitors. So did you consider transcriptomics when you sampled?
That is a really great question that I'm going to have to talk to you offline about, and bring in one of our Dr. Jason Lee and get talking that in more detail.
Great, thank you so much, Charles. Along the lines of the considerations that were discussed in Justin's presentation, have the potential environmental impacts of the cleaner and or decoding been evaluated?
So the coating itself was formulated in accordance with the Navy's environmental requirements, and the both federal and Navy, as described in the specification. There are materials. It is a qualified coating, and the inhibitor package that was introduced is a commercially available material that is on the currently used in architectural coatings and residential coatings for mold growth and prevention of mold growth. It is and it complies with all the Navy toxicity and metals requirements and limitations. So it was considered, and we made sure to stay within all federal and Navy guidelines.
Great, thank you so much to both of you. We're going to go ahead and start wrapping up. Before we do so, I'm going to pull Justin back into the discussion and ask you, Justin, and then Charles, if there's any key takeaway messages that you'd like to reinforce with our audience before we let you go. Justin?
Yep, thanks for the opportunity. Yeah, I would say, just like what Charles was showing with all those wonderful images of mold fouling every surface within the ships. I mean, the presence of fungal organisms requires water, and where there's water, there's life. And it doesn't matter at this point, it doesn't matter what coating they're landing on, they're finding a way to live. And the problems that we're trying to address in both a very fundamental and more applied way all lead to potentially addressing the environmental impact of the coatings that we create.
Thank you, Justin. Charles, would you like to add anything?
Absolutely agree. It has been a reaching out and interviewing ships force and doing this ship survey has been a interesting experience to see just how pervasive mold growth can be on different surfaces, and how how frequently, even after cleaning, mold growth can regrow, and how the concept of it's just a way of life and cost of doing business in these areas, and how the cleaning is the complete reactive method of controlling growth. And by Justin's work and the work that we're doing here, we're hoping to transition more long-term proactive materials to prevent this in the future. Again, we are always looking for partners and opportunities to demonstrate this coating on different assets and do different demonstrations. So any interest, please reach out and we would be happy to discuss.
Well, thank you both again for really great and very informative presentations and engaging Q&A sessions. We're going to go ahead and wrap up here with a reminder about the next webinar that's coming up on June 15, where it'll focus on autonomous underwater vehicle-based structural acoustic sonars for underwater buried UXO detection and classification. This webinar will feature Dr. Brian Houston from the U.S. Navy Research Lab. So please make sure to tune in. You can go to our webinar webpage and register for this webinar and other webinars through the beginning of next year. Before we conclude, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it this time if you can please take a moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you so much for joining us.